Hello everyone. Here we are going to present presentation on topic mitochondria and lysosomes. So let's start. In mitochondria, here food is converted into chemical energy called ATP. ATP is released by mitochondria so cells can use it. Mitochondria consist of two membrane. First, outer membrane separating from cytosol and inner membrane the surrounding the so-called matrix. The area between membranes is called inner membrane space. ATP is generated at inner membrane of mitochondria by inefficient mechanism called oxidative phosphorylation. Mitochondria are membrane-bound cell organelles that generate most of the chemical energy needed to power the cell's biochemical reactions. Mitochondria contain their own small chromosomes. Generally, mitochondria and therefore mitochondrial DNA are inherited only from the mother. Mitochondria's structure within the cell that convert the energy from food into a form that cells can use. The space surrounding cristae is a matrix filled with enzymes, coenzymes, water, and other molecules. Mitochondria are the sites of cellular respiration, which finally generates fuels for the cell's activities. Mitochondria are also involved in other cell processes such as cell division and growth as well as cell death. Mitochondria are semi-autonomous. They are partially dependent on cell to replicate and grow they have there. Mitochondria have a number of different shapes. A mitochondrion contain outer and inner membranes composed of phospholipid bilayers and proteins. The two membranes have different properties because of this double membrane organization. There are five distinct parts to a mitochondrion: the outer mitochondrial membrane and the intermembrane space, the space between outer and inner membrane. Functions: mitochondria are membrane bound cell organelles that generate most of the chemical energy needed to power the cell's biochemical reactions. Chemical energy produced by mitochondria is stored in small molecule called adenosine triphosphate known as ATP. Roles of mitochondria: production of ATP, calcium homeostasis, regulation of innate immunity, programmed cell death, and the cell regulation.
Hello here, my evolution of mitochondria. Mitochondrial DNA. Mitochondria are unlike other cellular organelles in that they have two distinct membranes and a unique genome and reproduce by binary fission. These features indicate that mitochondria share an evolutionary past with the prokaryotes, single cell organisms. Production: mitochondrial ATPase. The three processes of ATP production include glycolysis, the tricarboxylic acid cycle, and oxidative phosphorylation. In eukaryotic cells, the latter two processes occur within mitochondria. Interesting facts about mitochondria. Mitochondria are called the powerhouse of the cell as these cell organelles are responsible for producing ATP molecules, the energy currency of the cell. Mitochondria are a rod-shaped double membrane cell organelle with a distinct structure and specialized functions.
Hello everyone, I am here today to present my topic which is lysosome. Lysosome is a membrane bound cell organelle that contains digestive enzymes. They are spherical vesicles that contain hydrolytic enzymes. Function and structure. Lysosomes function as the digestive system of the cell. They remove worn out and poorly working cellular organelles by digesting them to make way for their new replacements. Lysosomes are formed by budding of the Golgi apparatus and the hydrolytic enzymes. Within them are found in the endoplasmic reticulum. Why are lysosomes known as societal bags? Lysosomes work as the waste discarding structures of the cell by processing undesirable materials and degrading them both from the exterior of the cell and waste constituents inside the cell. But sometimes the digestive enzymes may end up and damaging the lysosomes themselves, and this can cause the cell to die. This is termed as autolysis, where auto means cell and lysis means the disintegration of the cell by destruction of its cell membrane. Hence, lysosomes are known as societal bags of the cell. Thank
Discovery of lysosomes. Lysosomes were discovered by a Belgian biologist, Christian de Dove, and was awarded a Nobel Prize in Medicine or Physiology in the year 1974. He is also known for the discovery of other cellular organelles known as peroxisomes. Lysosome in plant cell. Lysosomes are predominantly found in eukaryotic animal cells and are responsible for breaking down cellular debris in plant. The role of lysosome in undertaken by the vacuoles as traditional cell biology detects. However, recent discoveries point out the function of vacuole is quite similar to the function of lysosome in animal cells. Clinical significance. Lysosomes are involved in a group of genetically inherited deficiencies or mutation called lysosomal storage disease (LSD), inborn error of metabolism caused by a dysfunction of one of the enzymes. The rate of incidence is estimated to be 1 in 5,000 births, and the true figure expected to be higher as many cases are likely to be undiagnosed or misdiagnosed.
Where are lysosomal enzymes made? Lysosomes comprise of over 50 different enzymes. They are synthesized in rough endoplasmic reticulum. Once synthesized, the enzymes are brought in from the Golgi apparatus in tiny vesicles or sacs, which then merge with bigger acidic vesicles. The enzymes produced especially for the lysosomes are mixed with the molecules monos 6-phosphate, making them to get fixed appropriately up into acidified vesicles. Lysosomal synthesis. Production of enzymes of lysosomes is organized by nuclear genes. Nuclear genes are genes which are located within the nucleus of cell, especially in eukaryotes. Any mutation in these genes may result in the emergence of over 30 diverse human genetic alignments, which are collectively called lysosomal storage disease. That means LSD.